Monday morning. YouTube is always slow on Monday morning. For those of you who have a paper, you can already share it in the chat. Okay. Well, welcome and good morning. Welcome to Neuroccino on the 8th of March. We're going to have a book review today about Alex Vanito's book about networks. And we have definitely one, maybe two papers. Let's see. Yeah, that's a yes from Victor. So the way it works is you briefly present your book or your paper, and then we talk about it to the best of our knowledge and just have a chat about it. And with that, I hand over to Eduardo for the book review.
Thank you. So I will try to share my screen. Let me see. Okay, so can you see the book or the PDF? Yeah. Okay, so I will start.
Maybe zoom in a little bit so we have the full page.
There?
Nice.
Yeah.
Thank you.
Okay. So first, well, hello, my name is Eduardo, and I'm doing a PhD here in the Neurocampus. I also host a board OT, which we recently started collaborating with the Neuroccino. So it's my first time here, and I'm very happy for that. And today I'll be talking about this book, which was very important for my master thesis. I did it in Amsterdam. And just this is the cover, so I don't have it on the PDF. And just to give you an overview of topics, you can find this book online on the Elsevier website. And basically, this book is one of the core books on network analysis in brain data. So they go through different metrics that one can compute, but also give a very interesting and critical overview of the methods and the history behind this sort of analysis. So it's really, really nice. It's not very long. And it's really good if you want to start working on network analysis, which was my case. I basically started working on fMRI data and graph theory, and I had no background on it. I've never really worked on it, and this book really saved my thesis, I would say. So just to give an overview of the book, so it starts with let me also these are the authors. So Alex Fornito, he's in Australia, Andrew Zaleski, and also Edward Wilmore. So these are the authors important to mention. Yes, so let me just go straight to the first topic. So on the chapter 1, they really go to the introduction of brain networks and why some people started looking on the brain data as a connectivity, or trying to understand the connectivity between neurons or neuronal populations or brain areas. So they explain how the brain is really complex, and we have to try to understand how it works and talks between different areas so we can understand functioning. And it goes through several data modalities and how you can try to infer this connectivity at different scales. So for example, at electron microscopy up to MRI and MEG. So gives a really nice introduction and to the topic, brings a lot of history on it as well, and also explains this idea of importing graph theory and from mathematics and physics to neuroscience. So yeah, a brief history on the graph theory, which is the main framework to understand brain connectivity. So it basically is the idea that you map the brain areas as nodes in a graph, and you try to create edges. You have edges between these nodes, and you try to understand how this is creating some specific properties of the network. Let me just see. So he really mentions some main findings that are well established in the field nowadays, that, for example, the brain seems to have this compromise between the local connectivity and clustering and some long paths to really improve how easy it is to get from one node to another, any other node in the brain, but at the same time having this compromise of cost, because if you want to try to have long wires in the brain, they are quite expensive in the metabolism test. So you want to have this system that is very, not really costly, but at the same time very efficient. And through evolution seems like this was what happened. That's what we call basically the small world networks, that this was discovered by using graph theory in brain data. And so they cover this main sort of findings on the field, which is nice. And they really have some really cool comparisons with other sort of networks. So for example, airplanes or social networks or maps, like this one in the image. So to have an idea of where else we could apply this sort of analysis, or where it came from as well, and to compare with different sort of networks in real life, let's say. Yes, so they really go, of course, into Ramon and Cajal, and how he basically started this whole idea of mapping neurons and trying to understand the circuitry in the brain at the micro scale. And then they really go through different methods one could use to understand this. So this, for example, is from MRI. There's another better image. Let's put it. So this would be for mesoscale connectomics from track tracing and viral injections, and they have some really cool images on it. And how, depending on the data that you have in the lab, you might want to consider different aspects of how to apply graph theory, because the graphs will be created in a slightly different manner. So I'll cover that now. So here in the chapter 2, he really establishes the basics, so the nodes and edges on the brain, and how you define what are nodes and what are edges. Of course, it changes across to depending on data type, but in my case, I was working on fMRI. So in fMRI, you have this image that will be registered to an atlas, and this atlas will establish the pool of voxels that you will have, and which will be the brain areas and where you have the signal. So each of these brain areas, we will consider them as nodes of the graph, and then the temporal correlation between areas are considered the edges. So basically, but for each data type is slightly different. And then, yeah, he goes into these details on establishing the different. And here, for example, he has this really nice image. They have this really nice image on the different scales and resolutions that you have for each method and data acquisition modality. And I think it's a very nice summary, more or less, on how to an idea of the temporal and size scales of these methods on the brain. For example, you have single cell patch clamp, but you also have PET imaging and fMRI data. Yes. Yeah, here he shows starting from a microscale, so electron microscopy. And I honestly find the images of this book so cool. So just to see the images is already really nice. Then to circuitry, more like a neuron-neuron connection. Then, of course, going more to circuitry and calcium imaging. Calcium imaging again. And then mesoscale, so more like the large scale track tracing, and then it goes into LFPs as well. So here. So yeah, connectome of a fruit fly. Quite cool. And then yeah, some different like LFP experiments you can do, and what sort of signal you can get from it, and expect to how to use graph theory. And then goes into, well, macro scale connectomics, so fMRI, DTI, diffusion tensor imaging. And I really like this image. So here they compare this brain atlas that we use in imaging with like how to divide our world map, and how much you lose or gain by dividing it more and more, or less and less. It's a nice comparison, because that's basically what we do with the data. We establish what are the areas that we want to look. And this can, of course, have a bias on your results. But even though they could find the properties of small worldness across any parcellation, so that's cool. And this is DTI, so diffuser transfer imaging is basically the flow of water and the structural connectome of the brain, which is pretty beautiful images. And I think you worked that in the lab, right, Stephanie? So it's quite nice. And yeah, so they go through all the methods, so it's really complete. It's good for a lot of people. And of course, then it's functional connectivity. It's more my side and where I focused a bit more. And then basically each chapter will be a different step on the analysis. So here's how to create a connectivity matrices and the brain graphs. So the idea of you have this sort of, you either have a matrix that you keep the weights, so the values of the correlations, for example, in fMRI analysis, or you can binarize it and just become like, I want data that is above this threshold, so it's going to become 0 and 1s. And they discuss how this can impact in further analysis, because the metrics that are used can be a bit or not suitable, or have different explanations depending on how you build your graph. And yeah, I won't really show it in detail because connectivity matrix is, well, quite known, I guess. But yeah, so this sort of images try to understand the connectivity in the brain. And so we have these ways of doing it, basically. Let me just, and then it goes into the specific metrics. So for the degree of the node, so for example, to how many other nodes the one node is connected, or how, then it goes into the centrality. So let me just go to the centralities. So yeah, centralities. So basically how well connected the node is. So you can have different types of centrality, and one will basically say if the node is important for the flow of information, or it's a node that is key, like very well positioned to communicate to all other nodes. So they discuss all these types of centralities. They always show the method to calculate the formulas. And also if there are some like main algorithms, they always go back to this reference and also compare it to other possibilities of computation. So that's good. So you can also kind of like have an idea of what's the main way of computing things, but what else you could do for your own analysis. And of course, it's full of references that are really useful if you go deep into the reading. Yes, so let me here just just gonna jump a bit so it's not too long. And then so for each chapter they will have these metrics, and I just want to highlight some. So in this case they look for what we call rich clubs. So it's this idea that the node has some important, the brain has some important nodes that are more connected and more important than others. But usually what happens is that these nodes are also connected to the other nodes that are more important. So it's like the ones that are more important are closer to the ones that are also more important. And this feels like this idea of a core of information sharing in the brain. And this has been mainly founded in DTI. So there's a guy from Amsterdam called Martin van der Huffel, and he studied that a lot. And yeah, we can always, we can find that on brain data, and we could understand brain organization using graph theory and this sort of imaging modalities. And yeah, the basics. Then they go again to the small world. They go a bit deeper in the explanation, as I mentioned before. This compromise between the length of the paths and how local connectivity is divided. And then a part that I think is quite interesting is that before finishing the book, they really go into this idea that you have to, it's not only about computing what is happening in your data, but you also have to have comparisons to no models or some sort of randomized version of your data to really say that whatever you're finding is not just because you could find a lot of randomness. So, but it's not just getting your matrix and randomizing it. You have to keep some properties to really be able to compare things. So that, of course, brain data will be different than random, right? So we have to sort of make the analysis a bit more complex and well taught, and they really show how to do this. So I haven't really worked on this yet, but it's a very nice chapter on how to work on this sort of analysis and comparisons, and to really say that whatever you're finding in brain data is relevant and not just noise, let's say. And they also have a chapter on statistical connectomics. So they also explain the statistical side of the comparisons that one might do using this data. So for example, when you go to fMRI multiple comparisons, how to correct for that, what are the pros and cons of this corrections, how strict they are. They also bring some methods on how to threshold the matrix, if you can just do it as an arbitrary threshold, or you have some methods that are based on the statistics to do it, to back your choice. And I think it's a very, very complete book. So if anyone is interested in this sort of analysis, should really have a look. And this also was based, the work that we developed in our lab, that's now a computational tutorial on GitHub to try to compute this metrics following more or less this bug structure on fMRI brain data. So yeah, it was a really cool book. Nice. And just I just want to tell you about something else. So I was discussing with Stephanie last week, and we thought it could be cool to share with you. So I don't know if you ever heard about this website called PubPeer, but the idea is that some researchers had some journal clubs in their labs, and they wanted the discussion to be out there. And so for other people, but also the author to see in the comments. And they developed this website called PubPeer. So basically what you can do is that you can use a DOI here, and you can find a paper, and you can comment on other people's work after they've been published. So for example, let's just get an example here. So here you can see that the paper is here, the title, and then someone commented something on the figure. And then the author replied on what happened and why. So this is a very nice way to interact with the scientific community. And also it's been used a lot to identify fraud, because you can post it anonymously. So it's sort of a way of having some sort of conversation after the papers are published and with your peers. So I think it's really cool and could be useful if you have journal clubs in your lab and you want to implement. It's a really nice tool. And lastly, I would like just to mention that, yes, we have the Bordeaux Tea. So it's a journal club at the Neurocampus on open science. And if you are interested on that, we are meet every month. And I will leave the link on the chat. Thank you very much.
Thank you very much. A lot of information. Thank you. That's great. Just as an addition to the website you just showed us, PubPeer, it also links into PubMed. So you can see when you do your search on PubMed, which papers are commented on.
Yeah, so they have this extension that you can install, and probably if I look for this paper and it's on PubMed, it will basically put a flag. You should put a flag saying this is on PubPeer. Let me see if it's gonna work. Doesn't seem to be. Oh, it's here. So as you can see, basically with this extension, they will always flag which papers were already there. So it's a nice way to know as well. And if you want to see if your own work's there, perhaps it's a good way to see if people are talking about your work as well.
Lovely, thank you. Coming back to the book, any comments, any questions? I know at least one of you has read it as well. You're all happy with it? Michelle?
So, it's not really a question about the book, but do you think a bit, because usually graph theory analysis are in 2 dimension, but do you think we'll be able to add like a third dimension and start creating object or graph in 3 dimension?
A graph in
Typically, when the
3 dimensions.
problem gets a lot more complex and you need to capture a lot, more of the interaction between the nodes, instead of doing it in 2 dimension and having it flat, you can do it in 3 dimensions to be more representative of.
Yeah, I think that would be very important, and I think that's more or less the idea behind using either, for example, in Pearson's in fMRI data, using a 3-point Pearson correlation or something like that, and trying to really have the set of mathematical tools to build connectivity from the data, but you can really infer the connectivity in a strong based manner. Because for now, for example, we have this new framework called topological data analysis, which basically try to go in other dimensions of the data and try to understand connectivity through higher order interactions. So it's not only pairwise, but beyond that, so triangles, tetrahedrons. But we are still working on the connectivity matrix as it is. So the best I would say would be to develop these tools in which you can find values of true connectivity between more than pairwise relationships. And I guess we are still like in the mathematical framework for that. I'm not sure if you know of anything else that could,
I think you won him over with the triangles already. So just for clarification and also to advertise your fantastic work, so you basically created a script that follows the step described in the book of how to analyze your data. Did I get this right?
Yes, more or less. It was strongly based on the book. Some things we had to adapt either because of the Python package was a bit different and how they define the metric, for example. But in general, yes, I would say overall, some things we built ourselves because it's not really covered by the book. But the core part follows the idea of the book. Yeah.
Lovely. So if you could share the link in the chat for everyone, that would be great. And there's one more question from Valentina.
You just stole my question. Yeah, I wanted to ask you if the book provides examples and what's the preferential or the book's advice for utilization of some software or Python code.
Yeah, so the book itself doesn't really go into that. They talk more about like the algorithms and the maths and alternatives in that sense. But the thing that we built in Amsterdam is the idea, that was the idea, is to build a sort of resource where you can find where to compute these things in Python, how to compute these things in Python, but also some things in MATLAB, because in the lab we also work with MATLAB.
Okay.
So there's a list of resources on at least where to start looking for things. Yeah.
All right. Thank you.
Oh, questions coming up now. Victor, I think, was first.
Hi, thanks for the presentation. I was wondering, because I've seen a lot of papers about the the network aspect of the brain, in particular the mathematical aspect of the networks, and I think it's very interesting to understand how it works. And very important to look at. And I was wondering if there was some applications, in disease. I mean, some clinical applications of these kind of models that were existing today.
On my side, what I've seen in the lab, to be honest, I'm quite new to the field on the clinics much more, so I don't know if Michelle would have more comments on that, but what I see that people seem to be mainly looking out for sort of markers, and they try to use these metrics to try to identify differences that would allow them to somehow interfere in the process of treatment. So for example, in the lab that we work in Amsterdam, they work on cognition. So they use these metrics to try to understand what might be happening in the brain so that then they can tackle and find a way of changing things, right? So I don't know if it's using stimulation on the brain, magnetic stimulation. So for now, what I see is more like using as a marker, and then you can think about the treatment afterwards.
Okay, thank you.
Leah, you also had a question?
Yes. So first, thank you for showing the book. Like when I had a look at this book, I just jumped to the last chapter when there was the statistical framework. So I didn't see or I didn't remember all these nice pictures. And maybe I should reread this book. So thank you to show that. And I want to ask you, like the sensation that I had when I was like reading these books about networks is that like, wow, a lot of publication, a lot of things that have been made like in different fields. So it seems that maybe you have like look to everything. So what's new? I had this sensation that this field was explored maybe too much, because at some point there were like network, and if you look also to the rate of publications about this field, it like exploded at some point. And so I had this sensation and also this sense of a little bit so lost. So what's the novelty, what I can do? So I think that what you do, it's really useful, like the book for implement these algorithms, then it's really useful. And I want to ask you if you had these sensations. So it was like a really huge field and really that exploded years ago.
Yes, I think you can see that it's like a quite established framework. I think maybe for the past 10 years, I'm not really sure, but I would say so, and has exploded indeed. So my work in Amsterdam was not, was this, but the focus on is something called topological data analysis. So it's a new framework for understanding brain data through networks and connectivity. So I would say that graph theory is really important, but we might need something else now, at least a different perspective, a different prism, and topological data analysis going that way. So if you want to perhaps find something on where to go, if you asked, I would maybe go on that sense. Or of course, it's also always important to have different data, or now we're collecting more and more data in different resolutions, right? So we might still apply this framework on better data, let's say. But on framework level, I would say we might want to have a look on topological data analysis.
Yeah, I agree. That it's a really interesting new perspective. Thank you.
Lovely. I think we got all the questions. Thank you again for presenting the book and showing us the websites. Don't forget to share the link to your own website so people can have a look at it.
Sure.
Right.
Thank you for the questions.
We're moving on to the first paper. Let's see if we can get both in today. And we're coming back to our main theme of clinical work with Parkinsonian monkeys today. Laura, take it away.
Okay, so this morning I wanted to present this paper. It came out last week in Nature Medicine. And I found it to be very cool because it uses this exciting technique, which is the autologous transplant therapy. And it is used to try to see whether we can, with this method, alleviate the symptoms related to Parkinson's disease. It is a study that was carried out in monkeys. So we can start from the assumption that Parkinson's disease is related to the degeneration of dopaminergic neurons in the midbrain. And given this, the transplant technique has recently been very interesting for the treatment of the symptoms related to PD. Indeed, there are different studies that try to use transplant, for example, of fetal mesencephalic tissues in order to try to improve the symptoms of Parkinson's disease in animals. But the problem is that when you use to graft cells from one animal to another, there can be issues of compatibility. So the fact that pluripotent stem cells can be extracted from other cells of the same individual opens the possibility to use cells coming from the same animal, transplant them after they have been declined into the type of cells that we need. So basically, the study uses 10 monkeys, which I think is quite a big sample for non-human primate studies. And these monkeys were divided into two groups. So give me 1 second. Okay. So there was one group made of 5 monkeys, which were then declined into the autologous group, meaning that they would have received a transplant of cells coming from themselves, let's say. So basically, tissue cells skin tissue cells were extracted. Then they were converted into pluripotent stem cells, and then they were in turn converted into dopaminergic progenitors. And for the autologous group, they were re-injected within the same subject, let's say, while the allogenic group, which was made of other 5 monkeys, received the dopaminergic progenitor that were derived from another animal. The Parkinson-like disease, let's say the animal model for Parkinson disease, was made by using this substance that is MPTP. And what you can see here is due to the fact that the authors decided to do a nemi-Parkinsonian model, meaning that the injection of MPTP was made only in the right intracarotid artery. So the degeneration was caused only for one hemisphere. And then the motor symptoms that derived could be seen only in the contralateral part of the body. So the injection was made on the right side. This is FMS, it's a fine motor skill scale, meaning that they did this task to the monkey to see whether they were able to do fine movements. And the higher the value of this FMS, the worse the performance. So here there is the comparison with pre-MPTP injection and post-MPTP injection for the left hand and the right hand, and we can see that the decrease of the performance could be seen only in the left hand, given that we have the injection in the right side of the brain. So as I said before, they use this FMS scale for evaluating the fine motor skills. They also use this clinical rating scale, which on the other hand tried to see whether there were symptoms that can be traced back to the classical symptoms of Parkinson's disease, for example, bradykinesia, tremor, and impaired gait. So after the animals got MPTP, there was a gap of 2 years, and after 2 years, they received the transplant of dopaminergic progenitors in the midbrain. And what you can see in these graphs is that the 0 represents the time in which the animals got the cell transplant, meaning that the previous 12 months was a period in which they were already intoxicated with MPTP. So they already showed symptoms related to Parkinson's disease, because also for the CRS, the higher the value, let's say the more evident were the symptoms related to PD. So they have these, for both groups, they have these very accentuated symptoms of PD. And then after the transplant, the two groups differentiate. In red, we have the group which received the allogenic transplant, which more or less remained the same in performance. On the other hand, the autologous group started to show less and less Parkinsonian-like symptoms. This is more or less the same for the fine motor skills. So we see that only the autologous group shows an improvement in the performance. So moving on, we have this data coming from a PET study. they were looking for let's say the activity, the binding potential of a substance that binds to dopaminergic neurons, meaning that we can see from that if we have an activity of dopaminergic neurons. The first two rows are devoted to the allogenic group. In the first row, we have the pre-transplant condition, and in the second row, we have the post-transplant condition. And basically, we can see that at Bayrai, there is no difference in the activity. On the other hand, when we move to the autologous group, the other two rows, we see, as it is indicated in these from these white arrows, that there is a change in the activity of dopaminergic neurons, and the arrow indicates the site in which the graft of a dopaminergic progenitor was made. So maybe it's more clear from this graph. We can see in red, we have the allogenic group. In blue, we have the autologous group, pre and post transplant. The binding potential, which is kind of a proxy of the activity of dopaminergic neuron. And we see that there is a huge difference on this binding potential, which becomes higher after the transplantation, but only for the autologous group. These are data that are done with TH immunostaining, okay? So the animals, after 2 years from when they received the cell transplant, were sacrificed. And we can see here, the first image is for the allogenic group, that the regions in which there was the graft, and in particular, as it is zoomed here, the putamen, there are some cells, but compared to the autologous group, these cells are in a region that is very, very limited, and of which the borders are quite neat. On the other hand, for the autologous group, it seems like the cells have well integrated in the host. This also can be seen here. So we have more cells that derived from the transplant of cells for the autologous group compared to the allogenic group. And yes, what you can see here is that, as seen before, the cells for the autologous group are more, have a major extent, let's say, and the wiring is higher. Also, this graph here shows more or less the same. It investigates the distance from the graph sites, in which we can still see neurons which were transplanted, or the extent wiring of neurons that were transplanted. So compared to a control condition that is given by the side that was not lesioned, in which the neurite density is more or less the same, and across moving from the site of the graft, which is the 0 here. On the other hand, for the allogenic transplant, we have this decrease of the presence of these neurons quite near from the site of transplantation, while for the autologous group, we can see that the decrease is more gradual, and we can still have traces of these neurons also at quite a far distance from the transplant site. So what the authors propose is that the immune system of the animals that received dopaminergic progenitors coming from other animals, so that underwent an allogenic transplant, was not, let's say, it attacked the cells coming from another animal, and therefore did not allow for the complete integration of these cells within the system of the host. The authors also did a correlational analysis between the performance, let's say behavioral performance, and for this CRS, meaning the Parkinsonian-like symptoms, and the binding potential, that is what we have seen to, let's say, represent the activity of dopaminergic neurons. And what we can see here is that the higher the FMS, meaning the worse the performance for fine motor skills. So the worse the performance, the lower the binding potential. So the best the performance, the higher the binding potential, meaning that the higher the activity of dopaminergic neurons, the better the performance for fine movements. It was also shown the same for the CRS, meaning overall the symptoms related to Parkinson's disease. And also they did a correlation, they found a correlation between this binding potential, so the activity of the dopaminergic neurons, and the total number of the cells that were grafted. Meaning that more or less they represent the same. So the higher the number of cells, the higher the activity coming from the putamen. Yes, so this is basically all. The authors also propose a kind of a model in which they estimate the number of cells that need to be grafted within the monkey. And therefore, given the ratio between the brain of the monkey and the human, to try to see which is the number of cells which should be grafted in humans, in order to have an improvement of the performance, which stays around 50%, as it is seen from this study. So yes, another thing that they do was to try to see whether also mood disorders that were related to Parkinson's disease could be alleviated by these transplants. So I'll show you, I think this is, yes. So they have these different indexes of mood disorders, which are, for example, anxious pacing, lack of motivation, or self-injury behavior. And what the authors claim is that when you have an autologous transplant, there is also an improvement of the mood disorders, let's say, or depression-like symptoms. But I'm not very convinced about this, because in this graph, you see that the 0 is the time in which they receive the transplant. So the red one is the allogenic group, while the blue one is the autologous group. And they claim that they had, let's say, less depressive symptoms in the autologous group. But from what I see here, for example, the allogenic group already had less depressive symptoms before the transplant. So Parkinson's disease was induced, and then here they had the transplantation, but as you can see, we already have a difference between the two groups. So I don't know if you have any hints on this. I would be happy to know what you think. And that's all. If you have any question or comments, go ahead.
Lovely. Thank you very much. Are there any questions? Yes, Victor.
Hey, hi Laura. Thank you for the presentation. I just wanted to know if this is a very promising technique, and I wanted to know if there was any human trial that were on the way or maybe planned for the future, because this is still a very terrible disease. So the faster we have something that work or even kind of work, the better.
Yeah, the only thing that I know about this is something that I read here. So it is not the first time that they try to use the autologous transplant for treating the symptoms of Parkinson's disease. So there was another study that did this in monkeys, but also one study that did this in a patient with Parkinson's disease. But as it says here, the patient with PD showed minor recovery 2 years after autologous transplant. Something that maybe I didn't see it, but I think something that is missing is in this study is that maybe there is no explanation of why it may, I mean, let's say, what is the reason why there is this difference between previous studies in which the transplant didn't get the same results in monkeys, or why these didn't work in the human patients. So I really don't know what is this, because it seemed to work good in this study. I don't know which is the limit that there might be between this study and previous study. And of course, the application in humans is something completely different. So, yeah.
Okay, thank you.
Leah, you also have a question.
Yes, thank you, Laura. I was trying to understand how this transplant was working. So if it's so allogenic is from another monkey, another person. If it's auto is from yourself. Is it correct?
Yeah.
And how it
From
works from yourself? Like from
I
I never heard about this. Sorry, I don't have the basics.
Also for me was something like, oh, this is super cool. Basically, from what I got, they take skin tissue from the animal, and there are a lot of procedure. You can see it in this paper in the methods section. It's super complex, and I was not able to fully understand it. But they do some preparation, given that you eventually end up with these pluripotent stem cells, meaning that they are cells that then after you can, with other preparation, decline into, I think, whatever you want, whichever type of cells that you want to obtain. And what they did was to do this preparation in order to decline these pluripotent stem cells into dopaminergic progenitors. And then they, for the autologous group, they re-injected these cells, therefore coming from the same subject, within the monkey.
Okay, so that was the process. Thank you.
Lovely. Are there any other questions? Are you scratching or no? You have a question, Michelle.
I genuinely have questions. Thank you very much for the presentation. That was really clear. I was wondering, so is there any genetic relationship between the allogenic group and the autologous group? Are they like brothers? How much do they share? Because if the cell do not work as well in the allogenic than in the autologous, there might be because of individual variability, but I was wondering how much the genetic making was different. Is it mentioned in the text of, whether like it's a colony of brothers and sisters or,
They don't mention it. I'm not sure about that, but given the fact that when I don't even know whether the allogenic group received the cells coming from, let's say, a line of cells the other group of monkey. Maybe it was a line of cells that was developed for another reason, or maybe from another group of animals. I'm not sure about.
I see. Okay. It's like the graph suggests that it would be this way, and then you're like, oh, it doesn't work, but it's a different, or the brain is working differently, and it makes sense. But if they're genetically identical or very close, there is something more that might lead to this different that is coming through the development.
For
Cool, really cool studies. Very fun and exciting. Thank you.
Lovely. I don't see any further questions. So in the interest of time, we're going to briefly move on to our final paper, which we probably only have time for a paper pitch. So you got 5 minutes, Victor. Make this snap.
Yeah, it should be all right. I mean, I just review the paper very briefly. I don't have much expertise in that, so in that field. So anyway, I won't be able to do a comprehensive review of it. But it's a paper that I've heard of a lot in the last weeks. It was published, I think, last month in Nature Neuroscience, and it's about mini brains, or well, brain organoids. It's in particular, it's about the long-term maturation, as I say the title, of human cortical organoids, and how their genetic markers develop throughout their culture, and to reach even a postnatal state that are something that is similar to what you can see in the genetic expressions in postnatal brains. So it's very exciting. Just to recap a little bit, so brain organoids are like, if you take some stem cells, you take cells, you make pluripotent stem cells, like in the article of Laura, you make pluripotent stem cells with them, and then you put them in some specific culture. How do you say? Well, you put them in some specific chemicals so that they differentiate into a specific type of cells. And then if you let them in this culture, they can form 3D, they will try to make a three-dimensional organ, a bit like them here. They will try to make a brain. They will not manage to do it, but they will end up in some spherical organ-like clump of cell. Here, it's they differentiate the cells into cortical brain cells, and so you get how they call it, a human cortical spheroid, which is the brain's the brain organoid that they are looking at right in this article. And so they say that right now we lack the access to these kind of tissue directly for study. So these kind of organoids are great, but we did not know if it was possible to have organoids that were anything else than fetus like brain or something like that. So it really limited the range of question you could try to answer with these organoids. And here in this study, they let the organoid in their culture for up to, I think, 9 months, or a bit more than 9 months, to check, and they systematically check for different genetic markers to see how the cells were expressing their genes, and what kind of gene were expressing what kind of epigenetic marker they could see in the cells. And actually they managed to show that there are a lot of paths that are evolving very similarly to what we can see in a prenatal brain, with the same kind of direction in the gene expression, And this goes on to even postnatal brain state. So you have very, it's a very exciting piece of research, because it really shows that you can have something that is, if not exactly the same, at least similar to postnatal brains. And this expands the framework of what you can do with this kind of organoids, and what kind of research you can do with regard to the disease that you want to study. For example, usually it was really about the neurodevelopmental prenatal state of the diseases that were studied in these kind of organoids. And now you can see that with this kind of research, you can pinpoint exactly at what point you want to study the developmental state of the disease. For example, for schizophrenia or something like that, there are some genetic markers that are very specific and that needs to be met. And with this, you can pinpoint where exactly in the development you can do that, and you can that also open the possibility to see if we can develop further after birth, after the equivalent of birth, so the postnatal brain, if we can continue to study the development of the neural development of brains with some pathologies, and to try to study them further. Of course, there are limitations with the fact that for now, it really takes a long time to get to this point, like 9 months to get to a postnatal brain, so brain-like structure, it can be very long. I mean, for people working with monkeys, of course, it's not. They would say, yeah, we work with real animals, and it takes way longer. It's way more painful to do this. But this kind of technique is usually more accessible. And if every time you need to do a very long culture, it can be a bit of a bother. So they are trying also to, well, it's for the future, but they will try to get a way to move back and forth the time, the development, the time. How did you say that? The point in time where no, it's not what I want to say. The developmental stage of the organoid. They want to be able to go back and forth in time, in the developmental stage, so that you can study exactly a specific time in the development of a disease. This could also be used to model epigenetic aspects of aging, which is great because it's also something that is hard to study in vivo. And yeah, there are a lot of different applications and some caveats that remains, but I think it's a good advance that we have here. So I think we are running out of time. So yeah, I'll stop there.
Lovely, thank you. We got time for one question or comment, if there's anything burning. No, in that case, thank you all for coming. This is all we have time for. For everyone on Zoom, if you want to hang back and get your questions out of the way, you're more than welcome to do that for a few more minutes. For everyone else, especially everyone over on YouTube, have an exciting week in science, and we see you again on Monday morning. Thank you.